for those of you who were at the last talk, that was a wonderful introduction, because what we're going to do, what I'm going to do in this talk is do this really deep dive into the female reproductive system. And so in many ways, the female reproductive tract is the black box of reproduction. And for the vast majority of internally fertilizing species, all the interactions between the male ejaculate and the female reproductive environment happen in here. And so it's these interactions that are really underlying the opportunity for post-mating prezygotic reproductive barriers or post-copulatory sexual selection. And while the field has spent a lot of time kind of figuring out what the male contributions are, and primarily the seminal fluid proteins that are transferred, and these seminal fluid proteins do a lot. They initiate a variety of behavioral effects, immune system responses, metabolic effects in the female. They stimulate a whole variety of reproductive effects. We know surprisingly less about what the actual female contributions to this environment are. And if our interest is in these ejaculate-female interactions, it's really important to kind of figure out those female. Along with that, we know a lot more about the male seminal fluid protein, the rapid evolution of male reproductive traits. So male genitalia has been really described as widely diverse, as well as sperm morphology and seminal fluid proteins. And while there's been less work done on those aspects in the female, every time we look at these female reproductive traits very closely, we see that same trend: rapidly evolving female genes, rapidly evolving secretory structures, sperm storage organs, and female genitalia as well. And while what's really interesting is we don't really know much about what's driving the evolution of the female part, though a lot of the recent studies seem to be showing that it's linked to ecology, we do know that increasing evidence is showing that the female is a selective environment for the male. And so things like long sperm storage organs select for longer sperm in males. And this is similarly also in the biochemical environment of the female, is a selective force on sperm and seminal fluid proteins. But so the female reproductive tract is really understudied in this way, and in particular, in one way it hasn't really been looked at, is that it's not a single structure. It's actually a composition of 5 distinct tissues that have to function in a kind of complex and coordinated way that both involves responding together to mediate reproductive events, as well as creating microenvironments based on their different specific functions. So for example, we have 2 sperm storage organs, the seminal receptacle and the spermatheca. We have 1 glandular organ, the perivaria. All these structures here are epithelial. The bursa is the site of fertilization, and the oviduct plays a really important role in egg ovulation. And so what I've done is I've done gene expression of each of these tissues at 3 different time points: unmated, 24 hours, and 6 hours after mating, or 6 and then 24. And I am not going to talk about any of the post-mating responses, but if you're interested in that, please come find me later. We have some really interesting findings there. For the rest of the study, I'll be exclusively focusing on unmated transcriptomes. And what we find when we do a hierarchical clustering is these really nice clusters of each tissue together. This is based on average at these time points. And notably, we find, so we have the rest of the female without the lower female reproductive tract as an outgroup. We've also included the fat body. So there's a very tightly associated fat body in melanogaster to the spermatheca and the perivaria. And it's often been a concern that these might be a contaminating structure. So we've included them both so we can say reliably that things that we're finding expressed in the spermatheca and perivaria are coming from the spermatheca and perivaria. But we also have some really fun hypotheses about how this might be a potential reproductive structure that I would also love to talk with people about after. So we find that the epithelial structures and the glandular structures form 2 different groups, but that each is separated each tissue is has its own cluster as well. And so we've done a couple of different measures to try and figure out what are the molecular signatures of these different tissues. So one of those ways was to look at enriched genes, and we determined those as genes that had a higher log 2-fold and significant higher expression profile in an individual tissue compared to the whole female without its reproductive tract. So a couple of notable things here is that the transcriptome complexity, as measured by the genes expressed in each tissue, significantly varied across tissues, with particularly the fat body having a very reduced transcriptome complexity. And then those colored bars on top indicate the genes that are enriched for each tissue. And we were able to compare those enrichments to find genes that were similarly enriched across common tissue types. And so reassuringly, we find muscular ontologies enriched in the epithelial tissues, which have a lot more muscularized epithelium and have these conformational changes in response to mating, where we see genes such as those involved in the endomembrane system more highly expressed in the glandular spermatheca and paravaria. So we also then worked to distinguish each individual tissue from the others, and we did that using a measure of tissue specificity called tau. And what I'm showing here is a principal components analysis on the unmated female reproductive tract samples, showing those different clusters, and then correlating the principal component loadings with the gene expression differences to show you that PC2 loading is significantly correlated with differences between the paravaria and the spermatheca, and those colored points are the points that we identified as tissue specific with our analysis of tissue specificity with tau. So I have developed this really comprehensive atlas of the female reproductive tract, and the question is, what can that really tell us about the evolution of the female reproductive system? So coming back to what I mentioned earlier, is male reproductive genes are really rapidly evolving, and in particular, these are genes coming from the seminal fluid proteins which come from the male accessory glands. And these have a number of really important features. They're tissue specific, they're secreted, and they're highly male biased. And so with the data that I have, we can ask the same questions about female genes, ones that are tissue specific, secreted, and sex biased. And then particularly one of our hypotheses going in is that we would see similar elevated levels of the gene specifically expressed in the spermatheca and the paravaria, which are the glandular tissues of the female reproductive tract. So first we looked at a level across all genes expressed in the female reproductive tract, and we noticed that there was a very clear difference in the distribution of genes that are not secreted and those secreted. I'm using secreted as a shorthand to mean anything with a signal sequence annotation. So we needed to take those two factors into account because of the interaction between secretion and tissue specificity. And when we look, I broke tissue specificity up into a couple of categories, and that the evolutionary rate of these genes significantly differs based on tissue specificity category as well as secretion. And so namely, genes that are tissue specific at a tau greater than 0.8 and also secreted have a greater DNDS omega value than others. And so looking specifically then at the tissue specific genes, so those with a tau greater than 0.9, we're able to ask the question, do we see differences between the female reproductive tract tissues? And the answer is not as conclusively as we had hoped. Tissue was not significant, secretion was still significant, and we did see a significant interaction between tissue and secretion. So what that is telling us is that there is possibly slightly elevated evolutionary rates in the genes that are tissue specific to the paravaria, the spermatheca, and the fat body, which is an exocrine, so all these secretory tissues, but that generally we're seeing kind of similar levels across all tissue specific genes. One of the takeaways from that is that all these different parts of the female reproductive tract are secreting into the female reproductive environment and are thus kind of potentially available for interactions with the male ejaculate. So the next part of this question that we wanted to get at are what is the sex-biased interactions going on? In part because among the tissue-specific genes, we actually surprisingly identified a number of seminal fluid proteins that we weren't expecting to see expressed in the female. And as a reminder, this is in the unmated female, so these are not coming from the male, unlike our previous story. So I looked at, again, this is looking at the relationship between tissue specificity and sex bias, and these sex bias measures come at the level of the whole fly from the MOD ENCODE data that's available. And what was really surprising, we found kind of this expected relationship that greater tissue specificity related to greater sex bias. But what was really surprising was that the tissue-specific genes in the female reproductive tract didn't have more female bias. They actually had a greater expression of male bias genes. And this included 65 seminal fluid proteins. And out of the seminal fluid proteins that have been identified using proteomic methods, that's about a third of the 212 that have been identified. So that's a rather large number. Most of them are not expressed in a tissue-specific way, though we do see a couple that are. And so those kind of 3 dark dashes in the middle of that white are all seminal fluid proteins specifically expressed in the parovaria, and namely one of them has actually been demonstrated to play a role in sperm competition. And we thought that, so the other thing we noticed is that most of these are actually expressed kind of broad levels across all female reproductive tract tissues. So we wanted to be able to distinguish seminal fluid proteins that were specifically expressed in female reproductive tract tissues versus those that might have more broad patterns of expression more generally. So we looked at the pattern of seminal fluid proteins across the tissues available from ModENCODE. So that includes your kind of standard somatic tissues such as gut and carcass and head, as well as sex-specific tissues of ovary, testes, and accessory gland. And what we found is that seminal fluid proteins group nicely into these 4 primary clusters based on their expression profiles across these tissues. The first and the fourth cluster, the 2 large top and bottom ones, generally represent seminal fluid proteins that are restricted, whereas or narrow as I'll use later, whereas the middle band are ones that are kind of broadly expressed. Almost all of the broadly expressed ones show up in the female reproductive tract, but it's actually a fair number show up outside as well. And we were wondering how that affected the evolutionary patterns and genomic distribution of seminal fluid proteins. So what we found is that it's the expression pattern across general tissues that influences the evolution of these seminal fluid proteins, not their expression in the female reproductive tract. But that expression in the female reproductive tract is what does contribute to the influence of genomic distribution. I'll just say we did find a couple of female bias genes. We don't know much about them. There's a number of them on the X. There's a lot of serine proteases. Most of them we don't know what they do. There's a number of rapidly evolving. We're excited to figure that out with some functional analyses. Takeaways, female reproductive tracts include rapidly evolving secreted tissue-specific genes that may represent critical female contributions to the reproductive environment, and that this is also a really interesting kind of finding that might reflect on the evolution of sex-biased expression, and these seminal fluid proteins can represent 2 different hypotheses, either the one that we're kind of really mulling over, this molecular continuity, that the female has to support sperm just as the male reproductive tract has to support sperm, so they're using the same genes to do that. Alternatively, this could be an example of conflict of male or co-option of genes in the female to kind of bias her reproductive events towards his optima. So just thank everyone in my lab, my funding sources at Syracuse University, which is on the Onondaga Nation land. There's a poster tomorrow on same-sex sexual behavior evolution, and I'm also starting to look for postdocs for next year, so come and find me later. Yeah.
So did you see any enrichment for female biased genes on the sex chromosomes?
No, so across all female biased genes, they had an equal distribution. There was also overall not a higher evolutionary rate, but there were some kind of interesting individual ones.
And you don't talk.
Thank you.